Good evening. Welcome to SpectraCell's 2015 webinar series. Thank you for attending tonight's webinar, Saving the Mitochondria. At this time, I would like to welcome Dr. Bernarda Zenker.
Thank you, Jessica. It's a pleasure to be with SpectraCell and the SpectraCell audience that are listening. Tonight I'm going to be talking about saving the mitochondria, which is a topic I'm passionate about. And in this next hour, what I hope to review with you in terms of the mitochondria are the basics of why the mitochondria is so important to us, some of its structure and function, some of the threats to the mitochondria, and then most importantly, different interventions that we can take as clinicians to help others in saving the mitochondria. So just to begin with, I just want to do a small amount of review of normal physiology. And as you can see here, this is a picture of the mitochondria, and it's an organelle, very simply, found in almost every plant and animal cell that we know of, and it's the site primarily of energy production. It's involved in respiration, and even though they can vary in size and shape, they're rather small little things, and they're only about 1 micron large, typically, micrometer, and it's surrounded by a double membrane. As you can see here on this electron micrograph, there's an outer membrane, and then there's this folded structure in the center of the membrane, and those are the cristae in and out, and surrounded by the inner membrane. And this is the area where energy is made, energy for every cell in the body and energy for all of our activities. So when we look a little bit further at the mitochondria, what we see is in the anatomy of a cell that there are many mitochondria, there can be many mitochondria within each cell. Now, in this particular diagram depiction of a cell, the nucleus is right there in the middle, those purple wavy lines, as you can see, is the endoplasmic reticulum. Excuse me. But the orange elongated oval structures, those are the mitochondria, and that is where our energy production comes from. Now, and as I mentioned, there are mitochondria in virtually every cell of our body, and even many hundreds of mitochondria within some of the cells of our body. Here is a diagram depiction of the back of the eye, actually, of the retina, and the very small grayish ovals, there are dozens of them here. These are the mitochondria, and they're extremely important, and a lot of research is being focused on studying and protecting the mitochondria within the eye as a possibility of preventing macular degeneration. The elongated structures at the top of the diagram, the purple and greenish blue teal diagrams, those are the rods and the cones. So if you go just below that, you can see in the gray matter of the choroid plexus here, these little oval gray structures, dozens and dozens of mitochondria. The thought is that if the mitochondria become damaged, dysfunctional, damaged, and die out, when sufficient numbers die out, the retina can detach and can lead to permanent blindness. And indeed, macular degeneration is the number 1 cause of blindness right now in our aged. And so there's a lot of research being put forward in how to preserve the mitochondria. And it's not only the mitochondria of the retinal cells, but in neurodegeneration, if the mitochondria die, then the cell dies. And we know that when looking at disease processes such as amyotrophic lateral sclerosis, Lou Gehrig's disease, and some others. So it's of paramount importance. to protect the mitochondria for the livelihood of the cell. This next slide that we're going to look at more closely is the mitochondrial structure and function. And as you can see, and it looks like a brown elongated hot dog almost, but when you open up that outer membrane and you look inside, you can see the cristae, the teal blue cristae, and the inner membrane structures. And when we look at the actual function, the functions of the mitochondria are actually listed here. That's why I've included this slide. The calcium channels are particularly important in the mitochondria. Mitochondria will store calcium, and calcium is needed for the actual respiration and the major functioning of the mitochondria to produce energy. There are multiple other functions that the mitochondria participate in, including detoxification of ammonia, particularly in the liver, production of heat, and when the mitochondria become dysfunctional, there's a whole process where apoptosis and programmed cell death occurs. All of this helps for cell longevity, as will be seen. When the mitochondria become dysfunction, the body has to get rid of those mitochondria in order for the cell to continue its livelihood and to keep to stay healthy. So it's very important just to understand this basic concept. If the mitochondria dies, the cell dies. And that's why I'm so interested in trying to protect the mitochondria and teaching others that we need to go to great lengths to know how to promote mitochondrial health, and then how to get rid of the damaged, more ill mitochondria within our cells. So tonight we're going to be talking about the dysfunctional mitochondria and how we get rid of them, how to support the good mitochondria, how to increase good mitochondria, and then also how to protect ourselves from further damaging of mitochondria. So when we talk about dysfunctional mitochondria, there are 5 major factors that are thought to contribute to dysfunctional mitochondria. The first is, and very importantly, nutritional deficiencies. And for this, it's nutritional deficiencies intracellularly within the cell. And, here at SpectraCell, we test those using the micronutrient test, and that is why one of the reasons the micronutrient test is so critically important is to understand the nutritional deficiencies that are occurring within the cell itself, not the deficiencies necessarily in the serum or in the plasma, but in the cytoplasm, in the mitochondria, so that they can be corrected to improve the health of the mitochondria. The second area that we've come to appreciate that will cause increased mitochondrial stress and dysfunction simply are having too many carbohydrates, too much fructose, carbohydrate and fructose excess, eating too much, too many carbohydrates. The third are infections and microbes and which would lead directly to dysbiosis. I would put into that area too of microbes and dysbiosis, not to underestimate mycotoxins potentially being exposed to mold biotoxins influencing and impacting the functioning of the mitochondria. Fourth are xenobiotics. Those are environmental toxins that we may not even be thinking about that are contributing to the dysfunction of the mitochondria. Sulfites accumulating in the transsulfuration pathway, not being handled sufficiently and cleared out, and many other influencing factors such as antibiotics, medications, things like this. We just want to raise our awareness that all of these can impact the mitochondrial function and lead to increased oxidative stress. And the thought now is that this oxidative stress influences the mitochondrial DNA, and that when the mitochondrial DNA has sufficient mutations, it's the mitochondrial DNA then will lead to mitochondrial dysfunction. When there's sufficient oxidative stress in addition, inflammation occurs. When that inflammation reaches significant height, then there can be failure of autoregulation and lead to inflammation dysfunction. So when we look at these 5 factors, it's most important to be thinking how to prevent these influences, because these are the 5 areas: the nutritional deficiency, carbohydrate excess, microbes, mycotoxins, other toxins, xenobiotics, oxidative stress coming in, causing the mitochondrial dysfunction that leads to further illness and compromise of the mitochondria that can then lead to cell death. So we move on specifically to treatment. When there is a buildup of dysfunctional mitochondria, and it may be due to antibiotic usage, for example, or medication usage, or coming into contact with pesticides and herbicides, how do we get rid of our dysfunctional mitochondria? Well, there are 2 things that we can do. Exercise has been shown to be able to deplete dysfunctional mitochondria. Exercise will help to eliminate or cull out the dysfunctional mitochondria through self-protective mechanisms. And what's advocated are short, intense bursts of exercise to be most physiologic and most helpful, using a variety of different exercises instead of one long marathon. Specifically for mitochondrial dysfunction treatment, seems to be more efficacious than running 5 miles nonstop or a 26-mile marathon. Doing a variety of exercises with short, intense bursts of activity will help mitigate, decrease dysfunctional mitochondria more quickly than doing a 2-hour run, for example. So when we look at this also, the second area to concentrate on in treatment of dysfunctional mitochondria is carbohydrate restriction, otherwise known as intermittent fasting. There have been reports that show, as this particular abstract states, 1 day of nutrient deprivation, such as fasting, suffices to halve your mitochondrial mass in various tissues. That's an amazing statement, that by fasting you can decrease your dysfunctional mitochondria by half. And intermittent fasting may invoke drastic recycling of the mitochondrial pool while improving longevity, even more so than continuous caloric restriction. So intermittent fasting, and patients frequently ask me, what do I mean by that? And I say, if you can do an intermittent fast, a 24-hour fast, 2 days a week, that would be more healthy than if you fasted for 5 to 6 days nonstop. So intermittent fasting will help to decrease the dysfunctional mitochondria, as does short, intense burst of physical activity, of exercise. This next slide shows the actual abstract where it was discussed that calorie restriction will increase muscle mitochondrial biogenesis in healthy humans. And as you can see from this very last slide, the very last sentence here, under the conclusion, the authors state, caloric restriction improves mitochondrial function in young, non-obese adults. So again, this intermittent fasting is so incredibly helpful for preserving and helping the mitochondria. So when we look at the mitochondria, this diagram that is now up, what I want to point out is we'll start in the upper right-hand corner, where you see the actual mitochondria, the purplish oval, and with a little square that points out where the mitochondrial intermembrane space is, and then the gray arrow coming down and a fuller depiction of this intermembranous space. And as you can see here in the center of the picture, where it looks like an accordion with globs of mud scattered throughout the accordion, what those globs of mud really are they're different complex channels, complex 1, 2, 3, 4, and 5. And at the bottom of 5, you'll see where ATP is coming out of complex 5 and being generated. This is what we're talking about as particularly interesting. What we want to know is how can we facilitate the mitochondria health in order to produce utmost ATP. And as you can see here on this slide, ATP is coming out of complex 5, and that is what we're interested in. So from this discussion, we're going to go backwards now and review how do we get to this point. There are multiple different interventions that we can advocate with our patients to be able to increase the supply of the needed nutrients in order to generate the ATP out of this electron transport chain. The accordion file here, where you see multiple different molecules moving in, moving out, the NADH moving in, complex 1, FADH moving in, complex 2, hydrogen ions into complex 3, and so on, getting to where we have an increase of ATP there at the end of complex 5. So now what I want us to do is go to the individual components, and I'm bringing up this next diagram that talks about acetylcoenzyme A. There are three ways that we know acetylcoenzyme A can be formulated, and it can come from proteins, carbohydrates, and lipid digestion. For each of these three ways, the proteins, the pyruvic acid from carbohydrates, and then the fatty acid, lipid digestion, there are different cofactors that are required in order to get those food substances that we consume in our diet into acetylcoenzyme A. So I want to start here on our right, talking about fatty acid synthesis. You have to have carnitine and magnesium in order to generate acetylcoenzyme A from lipids. If we don't have carnitine, sufficient carnitine and magnesium available, then acetylcoenzyme A will not be formed from the lipid digestion. Similarly, I'm going to go to our next slide. What you'll see is that in order to obtain acetylcoenzyme A from carbohydrates in the glycolysis pathway, you must have lipoic acid, vitamins B1, B2, and B3 in order for them to be metabolized appropriately and to have receive energy sources from them. Similarly, in the third aspect of where we get energy from proteins, you have to have zinc and oxygen. So as you can see, just in general here in this slide that I've put together, the nutritional cofactors in order to get energy production from proteins, carbohydrates, and lipids, we must have sufficient zinc, lipoic acid, B1, 2, and 3, carnitine, and magnesium. If these are deficient, then we're not going to be able to harvest or to produce as much acetyl coenzyme A as possible. And when we go to the next slide, you will see the acetyl coenzyme A is what's needed to go into the citric acid cycle, and this is where the energy is produced. Incidentally, I'd also mention on this slide here, it's this acetyl coenzyme A, for example, that is required for cholesterol synthesis. If we don't have acetyl coenzyme A sufficient, then our body is not going to be making sufficient cholesterol and steroids, the downstream products of your cholesterol synthesis, your progesterone, estrogens, testosterone, DHEA, etc. And in addition, here you can see acetyl coenzyme A goes on to the forming of the ketone bodies as well. So the take-home slide in this particular, the take-home message in this particular slide is the forming of acetyl coenzyme A from proteins, carbohydrates, and fats are dependent upon these cofactors. We can enhance our energy production by making sure that we have adequate carnitine, for example, or if an individual is overweight and trying to lose weight, it would be helpful to make sure that that particular individual has sufficient carnitine and magnesium to facilitate fatty acid, lipid metabolism and digestion. Now, just remember what I said about acetyl coenzyme A. This is a different diagram, but we're still talking about acetyl coenzyme A and coming in to make energy production. On the left, the upper left, you can see acetyl coenzyme A feeding into this cycle, the citric acid cycle, as one of the nutrients coming in, and then out of this particular cycle comes ATP in the far right-hand corner. If you look around the cycle, there are multiple areas that you see. NAD is coming off as well, and NADH in the mauve-colored little square boxes. This is critically important that we have the ability to make NADH, because the NADH is what feeds into the electron transport chain. So as a summary here, these fats, carbohydrates, and proteins, if there are sufficient cofactors, will feed in to the citric acid cycle, will feed in the acetyl coenzyme A into the citric acid cycle in order to have energy coming out the other side. This is the pathway, and this is how we can influence energy production, but in the mitochondria, by making sure that there are sufficient of these cofactors. And this is what I mentioned just a moment ago, that in this citric acid cycle, what is so important to understand is that this acetyl coenzyme A feeds into the citric acid cycle here, and the output is the NADH. The NADH is what's going to be going back into the electron transport chain, and I'll be showing that again in just a moment. When we look at the micronutrient test results that SpectraCell does, it's the areas that I have outlined here in red are the areas that are so important when we're looking at these cofactors of how to nurture and nourish our mitochondria. And the B vitamins are in the upper left-hand box here, the B1, B2, B3, so important for the metabolism of carbohydrates, very, very important. The lipoic acid here also under the individual antioxidants in that middle row, highlighted singly by itself over to the right, is lipoic acid. And then on the bottom row, the minerals that are needed for acetyl coenzyme A production, particularly magnesium and zinc, making sure that there's sufficient intracellular nutrients available. So when we look at all of this, again, your fats, carbohydrates, and proteins are supplying the nutrients that are needed for the body to make acetyl coenzyme A. And what is most important is that when this acetyl coenzyme A is produced, it feeds into the Krebs cycle, that round circle of multicolored ovals here at the bottom, the Krebs cycle. The important byproduct of the Krebs cycle are the NADH molecules. And as you can see, NADH is spiraling off, and there are dotted lines going up to complex 1. NADH is required in order for the electron transport chain to be functioning. So NADH being produced and supplying hydrogen ions to the inner mitochondrial space of the mitochondria. And if you look closely at this mitochondrial electron transport chain, what you will see is that in complex 1, 3, and 4, you can see hydrogen ions with being taken up into the inner mitochondrial space. But then in complex 5, the hydrogen ions come back through the inner mitochondrial membrane, out of which is produced ATP in that last royal blue image of complex 5. That's where the ATP is produced right here that energizes our cells. So it's the NADH being formed in the Krebs cycle, providing the hydrogen ions through the electron transport chain, of which then the hydrogen ions come back in complex 5, out of which comes our energy, our ATP. My little red circles here are a little bit off. I just am pointing that out where the NADH molecules are being generated and being sucked up, so to say, in the mitochondrial electron transport chain to produce the energy. In a summary slide here, what I've written here are in a chart form the support that is needed for the Krebs cycle, for the fat, carbohydrate, protein metabolism, and then for the citric acid cycle, the different cofactors that are needed for the citric acid cycle to be functioning in a healthy way. In this next slide, again, the Krebs cycle here, another depiction of it producing NADH. And in this particular depiction, they also show FADH. FADH, as you can see in the drawing to the right, a different presentation of the electron transport chain. You can see that NADH is absorbed up into complex 1, but FADH is what is absorbed up into complex 2. Both of these, NADH and FADH, give up hydrogen ions into the intermembranous space, and that's why it's so important to be having the generation of these hydrogen ions through FADH and NADH in the Krebs cycle. And if by chance, for some reason, your Krebs cycle is shut down, perhaps by heavy metals, mercury, arsenic, antimony, for example, fluoride and aluminum, can shut down the Krebs cycle. If that occurs, then it's imperative to be able to supply some of the NADH directly to the electron transport chain to bypass those blockages. And that's one of the therapies that I do recommend to patients if they have heavy loads of heavy metals blocking, to, as we get rid of the heavy metals out of the body, to be able to support the mitochondria directly with some type of NADH support so that this electron transport chain can continue and energy can be produced there out of complex 5. So this is, again, in a summation table of the different micronutrients that are required for these 5 different complexes to be working. And as you can see, for complex 1, niacin, riboflavin, iron, CoQ10, melatonin are listed here. That these are different micronutrients, and melatonin is a hormone, but these are things that can help complex 1 function most efficiently. So on a micronutrient test, for example, I would be very, very interested in looking at an individual's riboflavin, niacin, and CoQ10 levels. This riboflavin and CoQ10 is also needed, as you can see, in complex 2, the CoQ10 in complex 3 again. Complex 4 is interesting in that you need copper and vitamin C, and then in complex 5, the magnesium. All of those things and deficiencies can be evaluated and advocated if there are deficiencies that show up on the micronutrient panel. So what happens if your cells are on fire, however? So what we've looked at so far is, what do you do with dysfunctional cells? You exercise or intermittent fasting. How can you support the production and the function of healthy cells? And we've talked about the micronutrients. But now what I'd like to review briefly are some causes for cells being on fire. And some of the things that we know of that can do this are intense oxidative stress, insomnia, for example, exhaustion. Individual that burn the candle at both ends can do it. And then toxin overload, heavy metals, drugs, chemicals, mycotoxins. And there's a beginning field of interest in electromagnetic fields having direct effects on those calcium channels that we spoke of briefly in the mitochondria. So these are different things that we need to be aware of, can be affecting our mitochondria, and take preventative precautions. So if a mitochondria is distressed, we start thinking, what are some of the things that this stress can cause? And as you can see here, there's a whole slew of problems that can develop if we don't take care of our mitochondria. And as I said earlier, as I mentioned, if the mitochondria die, the cell dies, and the process between healthy mitochondria and cell death evolves with significant pathology in the intervening space. In cancer, obviously, diabetes, gastrointestinal distress, psychiatric problems, cardiomyopathies, obesity is now, there's a lot of attention being paid to the triggers that are causing obesity. So when we're looking at mitochondrial dysfunction and why things are not the way we would like them with our patients, we need to be thinking of primary and secondary reasons of perhaps mitochondrial dysfunction. And when we think about oxidative stress, it's burning the candle at both ends, yes, but there are many ways that oxidative stress can be occurring to the mitochondria. And through this nice little diagram, it points out several of these, for example, infections and microbes, but also too much sun exposure. Sun worshipers, as we know, their epithelial skin cells, etc., will react over time to too much radiation from sun, causing significant damage and then leading to cancers. Chemicals and environmental factors are very, very important. Not enough antioxidants to help squelch the stress going on inside the mitochondria, the reactive oxygen species. Too high of carbohydrate intake, eating too much, too many refined sugars, too much fructose, high fructose corn syrup will stress our mitochondria. So when we're thinking about oxidative stress, it's of course not getting enough rest, but it's also overeating carbohydrates. It's not getting the right kinds of foods, and it's these environmental stressors as well, the chemical toxins, the excessive sun exposure, and infections as well. So speaking about toxins, I just wanted to review this slide. Toxins can shut down the mitochondria. I love this slide because it demonstrates, again, the electron transport chain. Complex 1 here, where NADH is the very first depiction, this glob of purple, to the far left, where NADH is coming in, giving up their hydrogen ion. Well, if you look closely, it says rotenone here. Rotenone is Roundup, and Roundup can stop complex 1. That's very important for us to realize how devastating it can be. Here in complex 2, actinomycin A is a type of antibiotic. Actinomycin A can impede complex 2. In complex 3 here, as you can see, cyanide will shut off complex 3. Now, if we have sequential shutting off of our different complexes of the electron transport chain, then we're not going to be able to make sufficient energy. This cyanide that can shut off complex 3 is the cyanide, for example, that comes in cyanocobalamin. I encourage all of my patients to trash anything that has cyanocobalamin in it, in order to remove even if we can just remove part of the toxins that are shutting down the mitochondria, it will markedly help for longevity. So getting rid of cyanocobalamin, getting rid of pesticides, insecticides, and being aware that many of the drugs that patients end up taking will have direct consequences on our mitochondria. There was a very interesting article in 2013 that I reviewed titled "Bacterial Antibiotics Induce Mitochondrial Dysfunction and Oxidative Damage in Mammalian Cells." That was, and if you look here at the abstract, onto that third underlined phrase in red, bactericidal antibiotic-induced effects leads to oxidative damage to the DNA. Now, I'd like to show you a micrograph from that article of a cell prior to it being treated by ciprofloxacin. The far micrograph to the left is a solo, beautiful cell, and then it was treated with ciprofloxacin. 24 hours later, the electron microscope photo was taken, and you can see how the cell is disorganized and looks like it's crumbling, falling apart after 24 hours of ciprofloxacin. This was a very significant realization that our antibiotics can do nasty things, and directly from this particular journal article came the study ciprofloxacin plus N-acetylcysteine, NAC, was compared with ciprofloxacin only treatment. And what they showed was that there were similar levels of bacterial killing, suggesting that NAC does not interfere with antimicrobial activity. And then this last phrase, bactericidal antibiotic-induced oxidative damage, was significantly mitigated by the co-administration of NAC. This is a wonderful article that I just want to share with all of you, in that NAC, N-acetylcysteine, can help protect our mitochondria against the damage that can be done with many of our antibiotics. In this particular article, they tested ciprofloxacin. Now, we know ciprofloxacin is associated with Achilles tendon rupture, but on a cellular level, what this article suggests is that there is much more damage that can occur throughout the cells by affecting our mitochondria, and just by protecting the cell with a little bit of N-acetylcysteine, it would be so helpful. And this would be particularly true if on a micronutrient test, before giving ciprofloxacin, even we assessed the cysteine levels before adding insult to those levels, and just making sure that there's adequate NAC protection to protect our cells. This particular slide demonstrates this specifically, that N-acetylcysteine, NAC, helps to blunt the oxidative stress and inflammation that is directed directly to the mitochondria, because again, if the mitochondria die, then the cell dies and goes into apoptosis. So we have a great nutritional supplement, cofactor, that can help protect our mitochondria in N-acetylcysteine. This is just another depiction showing some of the other ways that the reactive oxygen species, it can be influenced and diminished. Again, NAC is there to the right as a component of glutathione that will help turn off reactive oxygen species. As you can also see over to the left, CoQ10 is a huge squelcher as well that we use to help fortify our mitochondria. CoQ10 and NAC are the two major nutritional supplements that can help protect our mitochondria. However, we also want to make sure that our mitochondrial enzymes, protection by mitochondrial enzymes, are sufficient, and there are two well-known enzymes that are critical for protecting the mitochondria. The first is the SOD enzyme, superoxide dismutase. There are actually 3 forms of superoxide dismutase. Superoxide dismutase 2 is there shown on the right, and it is the SOD that helps to protect the mitochondria, and it is manganese dependent. You must have manganese, sufficient manganese, in order to make SOD2. The SOD1 is in the cytoplasm of the cell and is dependent upon copper and zinc being present. As you can see from these 2 slides put together, the associated health problems that are thought to be occurring if there is deficient SOD. If there's deficient SOD1, amyotrophic lateral sclerosis and Alzheimer's are being implicated. If there is SOD2, this is thought to lead to premature aging and perhaps cardiomyopathies, neuronal disease, and then possibly cancer as well. So it's critical to have sufficient copper, zinc, and manganese in order to make these enzymes. And with this next chart from the Pathway Planner, I show where the SOD, right in the center of that red box, is where SOD is formed in the methylation pathway. And as you can see here, it is dependent upon zinc, copper, and manganese being present. If SOD is not being made in sufficient amounts, then the hydrogen peroxide and oxidative stress will further increase and cause mitochondrial dysfunction. So when I'm looking at the micronutrient test, I'm always looking here on this bottom panel of where the vitamins and minerals are stated, looking at how is the level of manganese intracellularly. And as you can see here, this particular report, that first analyte in the rectangle there to the far right is manganese, and her manganese, this particular patient's manganese, is in the borderline area. This is problematic because when there's intracellular stress going on, that manganese gets sucked into making more enzyme, then this borderline value is going to drop into the deficiency range, and then that person's going to be in a real pickle. And so you can look across here. Copper is also in the borderline, and so is the magnesium. This particular person is in this borderline danger area and needs fortification in these particular areas, or will get into problems. So this next slide shows glutathione. That is the second enzyme that I'm particularly always on the lookout for to make sure that a patient has sufficient cofactors and building blocks for making glutathione. Glutathione is selenium dependent. It's selenium dependent, and the building blocks for making adequate glutathione are cysteine, glutamine, and lysine. You have to have all four of these, or you're not going to be making glutathione sufficient in order to deal with the toxins and detoxification in phase 1 of the liver in order to get the toxins out of the body. So here in this slide, I've just put it together in chart form that most easily seen. The next area that I want to talk about for mitochondrial support is adequate phospholipids for membrane support. This particular depiction is of a macrophage, and it is the lipid bilayer of a macrophage, and what you can see are all of the different components of the lipid bilayer. What I'd like to point out is that all of the orange is phosphatidylcholine. If an individual is suffering from phosphatidylcholine, then the membrane support of the organelles and of the cell will over time begin to suffer. So we want to make sure that a patient has adequate phosphatidylcholine, and in the methylation pathway, I want to point out where that is being formed. In the upper left-hand corner of this slide of your screen, this biochemistry chart, this is a very simplified depiction of the methylation pathway. Phosphatidylcholine is formed here, and it is dependent upon the PEMT enzyme. PEMT enzyme must be functioning well in order to get adequate phosphatidylcholine being formed from phosphatidylethanolamine. There are two reasons I am always interested in someone's choline level on the micronutrient test. It's because if we don't have enough phosphatidylcholine to make choline, then we're not going to have enough acetylcholine. And acetylcholine, as we know, is the neurotransmitter for memory. If we don't have enough choline, then I advocate supplementing in the diet and supplementing with nutritional supplements in order to be sure that there's enough choline present to make acetylcholine in order to prevent the memory loss of dementia. In addition, as you can see in this slide depiction, choline, this phosphatidylcholine, is essential for cell membranes, for the choline needed for appropriate signaling in cell-to-cell signaling and in the cell membrane structure, and in the transportation of fats for the prevention of fatty liver and atherosclerosis, for example. So I'm always looking at choline also as a component for supporting our membranes and cell-to-cell communication. And on the micronutrient test, choline is evaluated right here on the center panel in this red, where the red triangle is. That's the choline evaluation. if this is low, that is a really big red flag, and I can, bet that an individual will have some perhaps memory impairment, fatty liver, or just pain all over the body because of the impact of low choline on cell membranes and on the organelles themselves. So in talking with patients about, well, where can they get choline, this is a very nice chart on the sources of where you can get choline in food sources. So beef liver, as you can see, and raw eggs, for example, are some of your better sources of adequate choline to help as food sources for our membrane support. So to enhance mitochondrial production for the replication of mitochondria will actually go through a meiosis and split and self-replicate. One mitochondria will split into two mitochondria, etc. I've listed here five of the supplements that have provided support in enhancing mitochondrial self-reproduction, and quercetin, resveratrol, branched-chain amino acids, grapeseed extract, and of course our exercise. So this is right here at my last slide, and I've just made a summary slide here of all of the different ways that we can support our mitochondria and protecting the health of the mitochondria, as well as getting rid of the dysfunctional mitochondria, making room for new healthy mitochondria for our energy sources of our body. Carbohydrate restriction, intermittent fasting, L-carnitine, lipoic acid, CoQ10, vitamins B1, B2, B3, thiamine, riboflavin, niacin. Our minerals are magnesium, zinc, copper, and selenium. Don't forget selenium is required for glutathione production. The building blocks for glutathione are cysteine, glutamine, glycine, and then the quercetin, resveratrol, grapeseed extract, branched-chain amino acids, the phospholipids as well. So this concludes my talk, Jessica. I'm here at the end, and I just want to thank everyone for listening.
Hi, Dr. Zenker. Do you have a few minutes to take some questions?
I sure do. If anyone has a question they'd like to present, that would be great.
Okay, sure. Our first question is, during the intermittent fasting, how much water do you recommend we drink during that 24-hour period?
As much as you can. As much as you can. At a minimum, the guideline, at an absolute minimum, day in and day out, the guideline that I have is I look at a patient's weight, and if a patient weighs 150 pounds, I divide that by 2, and that's 75, the number 75. So 150 divided by 2 is 75. A person should be drinking 75 ounces of water per day at a minimum. During the time of fasting, you really want to hydrate yourselves and to be moving waste products out through the kidneys into phase 1 of the liver, getting things into the bile and back into the stool, etc. So you really want a lot of water to be helping that. And I would encourage, because this is a fasting state, to really watch your electrolytes as well. You don't want you don't want to drink so much water that you go into a hyponatremic state. And so if you can drink electrolyte water, sugar-free electrolyte water, that's going to be the best kind of fluids to be taking in, at least some electrolyte fluid containing. But it wouldn't be too much to say 100 ounces during that 24-hour fasting in order to flush out some of the byproducts. because you've got to get rid of the bad mitochondria out of your body, and that's a detoxification process.
Great. Our next question goes along with the intermittent fasting also. They would like you to review how you are defining it, how long, how often.
I encourage. I have some patients that do it every week for 2 or 3 days a week. I would recommend to do intermittent fasting. Something that is comfortable, and I give patients the guideline 2 days a week, and to try to go for at least 16 hours of calorie-free, a calorie-free state. If someone can make it to 24 hours, great. Many of my senior citizens cannot, and so I specifically tell them to do this. If they can, let's say they're going to do a fast on Tuesday and Wednesday. I have them get up on Tuesday morning and have a moderate breakfast, and then I will have them have a late lunch around 2 o'clock in the afternoon. So they have a late lunch and they're finished eating by 2 o'clock in the afternoon, say 2:30 in the afternoon. And that usually tides them over through the evening so that they really don't even get too hungry if they're drinking plenty of water. And then they go to sleep. So they're basically sleeping most of their intermittent fasting. If they, the next morning when they wake up, in terms of exercising, what I would like them to be able to do is drink fluids and exercise, and like a short burst of exercise for about 20 minutes, as long as they're not diabetic or have hypoglycemia issues, for about 20 or 30 minutes in the morning, and then they can eat. That would get them easily through a 16-hour fast. If they could wait until lunchtime, all the better. But if they can do at least 16 hours, that would be an intermittent fasting. And to do that 2 days per week. Some of my patients do it 2 days per week, every week. But at least a couple of times a month would be helpful, specifically for getting rid of old, burnt-out mitochondria. Is that helpful?
That's very helpful. It actually looks like we are out of time for any more questions. Thank you again, Dr. Zenker.
You are welcome. It's always a pleasure.
If you have any additional questions, you can still enter them in the Q&A after the webinar ends, and we will capture them. Thank you for joining us, and good night.
Thank you. Good night, everyone. Have a good evening.